Welcome back. Our first speaker in this last session will be Dr. Thomas O'Connor, who is the Director of the Division of Product Quality Research in OPQ's Office of Testing and Research and is a member of the CDER's Emerging Technology team. His responsibilities include managing research and testing projects that answer and anticipate pharmaceutical quality-related regulatory challenges through scientific approaches. We will also hear from Ernie Penachio, who is Vice President of Technical Operations at Continuus Pharmaceuticals and is responsible for formulation, process, and analytical development, technical operations, engineering, facilities, IT, and MS&T. Finally, we will be joined by Dr. John Lewin, Chief Medical Officer at On Demand Pharmaceuticals, where he oversees the quality and regulatory functions and is developing current and future manufacturing operations. Remember to enter your questions for the speakers in the Q&A chat pod, and we will try to get to as many as we can at the panel discussion after these presentations. Speakers, take it away.
Hello, my name is Thomas O'Connor, and I'm the Director for the Division of Product Quality Research in the Office of Testing and Research in OPQ. And it's my pleasure to be here today to talk to you about FDA's Advanced Manufacturing Product Development Science Program, part of our strategic initiatives to support the adoption of advanced manufacturing for pharmaceutical products. The learning objectives for today's presentation are, first, describe OPQ's capabilities when it comes to product development science and advanced manufacturing. Second is to list our current focus areas within advanced manufacturing. And the third is to use case studies and specific projects to explain the impact that product development science has on OPQ's mission. OPQ's Science and Research Program helps enhance FDA's capacity for evaluating and monitoring drug quality, safety, and efficacy. It helps modernize current regulatory pathways, which is very important when it comes to emerging technologies and advanced manufacturing. It can help address regulatory and scientific issues that are mission critical. It helps maintain a state of readiness that anticipates future needs, so the capabilities there ready when it needs to be used to address future issues. As you can see from the top picture, FDA staff have been in the labs throughout the pandemic to help address mission-critical issues, of course following the appropriate safety procedures to protect the health of staff. The bottom picture highlights another way that OPQ's Science and Research Program helps enhance FDA's capacity, and that's through training, where FDA staff can come to the labs and get trained on different technologies. Next, I'd like to talk about OPQ's research and how it interacts with CDER's Emerging Technology Program. Emerging technologies, by their nature of being novel, means that we have limited assessment experience with them being involved in drug applications. This means there typically can be open scientific questions, which can spur the need for research. The knowledge gained from internal and sponsored research can then help inform FDA's decision-making when it comes to policy, review, inspection, and surveillance activities, and ensure that FDA's regulatory policies reflect the state of art of manufacturing science. There's really a good feedback loop between the CDER's Emerging Technology Program and the research. When sponsors engage with the ETT early in their development, they typically will highlight key questions where there is uncertainty and they're looking for FDA feedback. As needed, sometimes these questions need research to advance the product development science to properly address them. This way helps align OPQ's research program with the needs to help ETT provide sponsors the feedback they need. And this creates a shared learning and open communication loop between both industry and FDA to help accelerate the adoption of emerging technologies and ultimately to advance product quality. OPQ's product development science capabilities encompasses many different areas. It includes novel manufacturing methods. That includes continuous manufacturing, 3D printing. It also includes areas in precision analytics that better help us understand the especially complex products that are made by these advanced manufacturing methods. We've invested in a number of high-resolution methods, including high-resolution mass spectrometry and NMR. There's also advanced manufacturing also takes advantage of inline monitoring tools to provide additional quality assurance. And these can be, spectroscopic, NIR, Raman, imaging. And we've also built up the capability about how we use this information, including advanced process control. We've also developed a computational lab that supports modeling and quantitative analysis, including physics-based modeling, CFD and DEM, to multivariate chemotrition analysis, and also newer machine learning methods. Finally, we focus areas in new advanced manufacturing that can support the making of complex products. This could be extrusion, as also emerging therapies like oligonucleotides, where we've developed capabilities both on the synthesis of the oligonucleotide, as well as sponsoring research that looked at the formulation and making those drug products. The intramural drug product development science program can be grouped in the following areas as we look across the projects that FDA has conducted or is conducting. We have a number of projects that revolve around novel manufacturing methods for small molecule drugs. Big area of focus in precision analytics. Another big focus area in advanced manufacturing biopharmaceutics. The sub-area that kind of really focus on the manufacturing of glycoproteins. And finally, a project in the manufacturing of synthetic nucleic acid sequences, and some projects that are cross-cutting in process modeling and artificial intelligence machine learning. Across these projects over the last few years, more than 65 internal reports and publications have been published to support communication of findings both to internal business partners and external stakeholders. The product development science program, though, also includes extramural sponsored research. This is because collaborations allow OPQ to leverage external expertise and capabilities to address scientific issues. We realize that a lot of these issues cannot be addressed by FDA alone, that we need to leverage the scientific community to help advance the science. We have multiple mechanisms to support collaborations, including OPQ U01 grants and FDA's Regulatory Science Board Agency announcement. Over here, we've highlighted some of the academic and small companies that we've collaborated with to advance different aspects of advanced manufacturing, whether it's continuous manufacturing of lipid nanoparticles or continuous manufacturing of small and large molecules, drug products. Right now, looking at the portfolio of projects, we have a number of projects in the Industry 4.0 smart manufacturing area, another group in the novel manufacturing methods for both small and large molecules, a group in novel process analytical technologies, process modeling simulation, and one project related around advanced manufacturing training. And earlier this year, over the last few years, these projects have generated more than 13 publications, and many more publications are emerging from these projects to support communication with the scientific community in these areas. Next, I'd like to highlight some specific case studies to highlight how the Product Development Science Program can support OPQ's mission. We'll look at examples across a number of different focus areas in advanced manufacturing. And I'll start with one example from precision analytics. First project area I'd like to talk about is the multi-attribute method. MAM is a liquid chromatography mass spectrometry-based method that's been proposed as an ETT as a replacement for conventional QC methods for controlled testing for therapeutic proteins. The figure shows a high-level overview of the method, first digesting the protein, initial analysis by LC-MS, and database searching to fully characterize the protein. And then in the control setting, LC-MS methods are generally used to quantify quantify CQAs, as well as for search for new peak detection. Based on our research and understanding of these applications, FDA published a perspective paper in 2019 outlining 4 major points to consider for QC implementation of MAN methods. First is the risk assessment. The benefits and risks should be weighed and may be specific to a product or certain CQAs, as this method may be more beneficial in some situations than in others. A method validation can be based on ICH and FDA guidances. LCMS methods may be more complicated, and certain aspects may need special attention, like LOD, LOQ, precision, and system suitability. New peak detection allows for detection of changes that are not directly monitored and should detect unknown impurities, and success of this feature is highly dependent on software parameters, including retention time windows, mass accuracy window, and peak detection thresholds. And finally, comparison with conventional methods. Comparisons may be complicated, as conventional methods tend to be less targeted than MAM, and analytes often will not directly correlate. Internal MAM capabilities have been developed using rituximab, along with other MABs, as a model protein. Internal assessment of the method is focused on the points to consider, specifically method validation, new peak detection, and comparison with conventional methods. This research has supported the evaluation of multiple ETT applications with MAM. And these applications involve products at different stages of product development. And for one applicant, ETT assessors agreed to a sunset strategy for conventional methods to be eventually replaced with a MAM-based method. Next, I want to highlight some projects related to novel manufacturing methods. The first is the continuous manufacturing of synthetic APIs. There's growing industry adoption of this emerging technology. There's been one approval of a high-volume drug substance manufactured by continuous synthesis, and there's been growing industry engagement with the emerging technology team. Both generics and innovator companies have started exploring the potential of continuous reactions to produce drug substances that can be difficult to manufacture by traditional batch processes. ATT is also engaged with sponsors looking to implement end-to-end manufacturing platforms that convert raw materials into final dosage forms, include both continuous synthesis and crystallization processes. Flow reactors can enable unique synthetic routes that may produce different impurity profiles. Continuous API manufacturing often involves fewer isolated intermediates and can contain telescope reactions utilizing innovative chemistries. It is important to understand the impact of these telescope reactions, where multiple reactions may be included in a single step on the generation and fate of impurities. And this understanding can also form the designation of regulatory starting materials for streamlined processes. OPQ has established a continuous crystallization platform for carbamazepine using a mixed suspension, mixed product removal system with PAT tools such as focused beam reflectance measurement and Raman spectroscopy for process monitoring and control. Building up this experience, and we are now including the study of continuous synthesis, an integrated process that combines both continuous synthesis and crystallization steps. An external collaboration led to the development of a pilot scale process, including a reactive crystallization step of a model API compound. This research has supported ETT feedback of sponsors looking to implement continuous API manufacturing, as well as support the development of ICH Q13. Unlike the earlier draft FDA guidance, Q13 includes both drug substance and drug product and has annexes for both synthetic APIs and drug substance and end-to-end manufacturing. 3D printing is another novel manufacturing method that offers an opportunity for precision medicine by being able to deliver and tailor production to different patient populations. Opportunities include developing a wider range of dosage without excessive alterations to the process and produce convenient fixed dose combination forms. Because this manufacturing process is very different from traditional tablet methods, the special and appropriate material selection and controls is one of the key ETT discussion areas for this technology. FDA has created a shared facility for 3D printing across the medical product centers that allows for sharing of expertise and resources. As an emerging technology, indicators of quality for materials and processes using 3D printing have not been fully established. One study conducted at the FDA looked at characterizing critical material attributes of semi-solid paste formulations filled into in cartridges for 3D printing. Pastes were characterized for viscoelastic characteristics, used during the 3D printing process, including creep recovery, cross modules, and extrudability models. Proper material characterization allowed the minimization of product quality defects. In another study, because 3D printing offers expanded flexibility in design of the dosage form, we developed physics-based models that were capable of predicting the hardness and dissolution of various 3D printed tablets. The dissolution models considered the swelling of the filaments and diffusion of the API. This opens up a mechanism to quickly design tablets for a given performance. These studies have supported the ETT feedback to sponsors, both in the area of material characterization and control of the process. Next, I want to discuss a couple projects related to modeling and quantitative analysis. For CM processes, understand the process dynamics in relation to material properties, equipment design, and process conditions is fundamental to understanding potential risks of CM to product quality, largely because of the potential impact on material traceability and disturbances, generation, and propagation. Such an understanding can be achieved by characterization of the residence time distribution. The RTD can be used to characterize axial mixing and predict how fluctuations in the feeding can dissipate in a mixer and their subsequent impact on the blend and content uniformity, thus predicting the impact of material feeding disturbances on blend and content uniformity in real time. OPQ-sponsored research has focused on understanding the impact of the method of measuring the RTD on the accuracy of model predictions, such as tracer selection, and on the impact of material properties and process parameter variation on the RTD and subsequent model predictions. The figure shows a funnel plot, which is the output of these modeling efforts, which can calculate the impact of a disturbance on product assay. This research can then inform risk-based model validation and lifecycle maintenance approaches. OPQ has established a computational group in its laboratory, develop and use modeling and simulation, and has combined with laboratory capabilities to study RTD models. In addition, extramural collaborations have been established to conduct round-robin studies to validate RTD models across multiple manufacturing lines. These results have been supported the application assessment of RTD models and provide ETT feedback. It's also provide training simulations as part of an internal training class for FDA reviewers. Lastly, I want to highlight one project related to advanced manufacturing for complex products. Liposomes are a robust drug delivery system that have been developed into FDA-approved drug products for several pharmaceutical indications. Direct control in producing liposomes of a particular particle size and particle size distribution is extremely important since liposome size may impact cellular uptake and biodistribution. As part of an FDA-sponsored collaborative research project with a team of investigators at UConn, a device consisting of an injection port was fabricated to form a coaxial turbulent jet in co-flow that produces liposomes via the ethanol injection method. By altering the injection port dimensions and flow rate, a fluid flow profile, i.e. the flow velocity rate versus Reynolds number, was plotted and associated with the polydispersity index for liposomes. Research was able to demonstrate that certain flow conditions produce unilamellar monodispersed liposomes, and the mean particle size was controllable from 25 up to 465 nanometers. The mean liposome size is highly dependent on the Reynolds number and on the mixed ethanol to aqueous phase and independent of the flow velocity ratio. While the research has started with liposomes, it has expanded to other nanoparticles, including lipid nanoparticles and polymeric micelles. Over a course of 3 projects, the system has developed from a benchtop proof of concept to a mature manufacturing platform that includes both automation and online monitoring. The platform enables potential benefits that were observed in other areas for continuous manufacturing, namely reduction in processing time per unit dose, reduction in equipment footprint requirements, and potential flexibility in duration of manufacturing campaigns that allow better responses to drug shortages or emergencies or patient demand. So looking back and summarizing the output and impact of the product development science program across the different focus areas. The research has directly supported ETT feedback and application assessment for over 10 ETT projects. It supported policy and development guidance. It's informed the development of ICH Q13 for continuous manufacturing of drug substances and drug products. and is supporting the development and implementation of the Advanced Manufacturing Regulatory Framework. It's also supported workforce development by providing training and supported the ETT graduation of continuous direct compression. Moving forward, CDER is constructing a research manufacturing pilot plant that is set to be available in 2022. The pilot plant will increase FDA's capability to generate knowledge and train FDA staff. Plan to continue to invest in research programs, both intramural and extramural collaborations, for growing areas in advanced manufacturing. As an example, we awarded 5 new collaborative projects in September 2021. And we'll continue to align the research programs to support the Emerging Technology Program and the development and implementation of the Advanced Manufacturing Regulatory Framework. In summary, OPQ has developed product development science capabilities across several key areas in advanced manufacturing. This includes leveraging external collaboration to enhance OPQ's capabilities, realizing that we need to engage the scientific community to make as much progress as fast as we would like. Product development science programs are aligned to address mission-relevant regulatory and scientific issues and to support FDA's decision-making related around the assessment of advanced manufacturing used to make pharmaceutical products. And product development science programs have directly supported the assessment, inspection, policy, and guidance development for advanced manufacturing. I have some challenge questions. 1. Identify product development science focus areas for advanced manufacturing. Continuous manufacturing, precision analytics, process modeling, or all of the above. B. All of the above. Second question, which of the following statements is not true? Product development science program only involves internal FDA research. Product development science programs align with the implementation and needs of the Advanced Manufacturing Regulatory Framework. Product development science program has impacted quality assessment, policy development, and workforce training. CDER research manufacturing pilot plant will increase FDA's capability to generate knowledge and train FDA staff. A. This, I'd like to conclude my talk and take any questions during the panel discussion. Thank you.
Thank you for that introduction. My name is Ernie Penachio. Today I will be giving a presentation on end-to-end continuous manufacturing of pharmaceutical products. I will begin our presentation with a brief background of Continuus Pharmaceuticals. I will then describe a contract that Continuus Pharmaceuticals has received from the Department of Defense and Health and Human Services for the onshore production of critical medicines. I will then take you on a virtual tour of the integrated continuous manufacturing factory that is currently in the design phase and is part of the contract with the Department of Defense. I will then discuss a challenge that is associated with end-to-end production of continuous manufacturing and possible solutions, and then I will give final thoughts. Continuus Pharmaceuticals is a spin-out of the Novartis MIT Center for Continuous Manufacturing. This center was developed for the innovation of continuous manufacturing in the pharmaceutical space. The company is backed by major pharmaceutical equipment manufacturers, for example, EMA, which is a pharmaceutical equipment manufacturer. We are also backed by private and professional investors. Our expertise is in integrated continuous unit operations, reactions, crystallizations, extractions, filter and dryer, all your traditional drug substance unit operations, as well as drug product, OSD, vial filling, control systems required for continuous manufacturing, and process analytical technologies. Our projects range from generics to partnered programs with pharma companies in order to develop integrated continuous manufacturing processes from batch processes. Continuus received a contract on January 15th of 2021 for the onshore production of 3 medicines that are on the short list. The facility is being designed to produce drug substance and drug product. We are building in the capability of producing sterile vials as well as oral solid dosage forms. The oral solid dosage forms will be produced from our extrusion mold and coating process, which is proprietary to Continuus Pharmaceuticals. We are also designing the ability to utilize a continuous liner drum station in order to isolate API into drums, in addition to drug product. The facility will be a multi-product facility. The facility also will have the capability of managing, producing, storing, and handling potent compounds all the way down to operational exposure band 5. In addition, we will give the facility the ability to handle, manage, process, and distribute DEA scheduled drugs. The design intent for the plant is a capacity of 100 million vials per year. So what does an end-to-end continuous manufacturing process train look like? This is a diagram of an end-to-end production pathway for sterile vials in the ICM factory. We start on the left-hand side in the dispensary. In the dispensary, we will dispense raw materials, intermediates, and precursors, mostly in the powder form. And we also have a tank farm where we can dispense reagents and solvents. Continuous API manufacturing takes continuous feeds from the dispensary and the tank farm to continuously convert raw materials into API drug substance. The API drug substance is manufactured continuously and is continuously collected in the API in-process storage area. The API in-process storage area acts as a buffer to allow for differences in capacity of up and downstream. Bulk API is then fed into the vial filler formulation area. This is accomplished by taking a bulk API out of the in-process storage vessel, creating a concentrated liquid, and then pumping the liquid into the formulation area for continuous dissolution and formulation of vial solution. Once the continuous dissolution formulation is produced, it will be continuously pumped into the vial filling line where sterile vials will be produced. In the vial filling line area, we have sterile vial filling, we have stoppering, capping. We then will progress to terminal sterilization, then through the vision system, finally to the bulk vial pack-off station, and into the GMP warehouse. In addition to aseptic vials, we can also produce bulk API. The API production process is the same. The difference here is instead of going into the API in-process storage vessel, we go straight into a continuous liner bag dump station where we can drum off API, and drums will then be placed into the warehouse. We are now ready to go from the pilot plant to the commercial production scale with the help of the contract from the DoD, and we would like to scale up our pilot plant, which you can see on the left, to the commercial plant which we have designed, which is on the right. We've selected 32 Cabot Road in Woburn, Massachusetts as the site of the ICM factory. The ICM factory is a 49,600 square foot warm shell, as can be seen on this diagram. This is the building right here, where you have the street, you have the entrance in the front, you have great access around the building, plenty of parking. You have a loading dock and an area for a truck to back into right here. This is what the current design looks like in detailed engineering. This is the site plan. The existing warm shell. This is a building addition that is required. The building addition consists of a combination of process area as well as office space. We also will be required to build a utility structure which will house air handlers, chillers, boilers, and utilities required for running the factory, as well as a tank farm for the storage of solvents and waste. This is a 3D view of the ICM factory. You can see on the back, you can see the addition. This addition is where we have process areas on the ground floor and office space on the second floor. We also have solvent storage in the back. This is the utility structure here. And we have also a tank farm in the back here. In addition to the addition we have on the back, we will be installing dunnage on the roof, and we will be placing a penthouse with all of the air handling equipment we need for the second floor up on the dunnage. All the air handlers for the first floor are located in the utility structure. The existing conditions of the warm shell are here. We have a central stack which has bathrooms and an elevator in the front, and in the back we have a freight elevator and an exit stairwell. This is the current detailed design of the ICM factory, the ground floor on the left and the second floor on the right. We'll start in the warehouse. This is a just-in-time warehouse that have the capacity of 2 to 4 weeks of raw materials in process and drug product. We will need a second warehouse for bulk storage of raw materials, intermediates, components, and drug product. The warehouse has controlled ambient storage, a DEA vault, and a 2 to 8 Celsius cold room. This is the API production area. The API production area consists of 2 separate suites. The first is the high volume suite. The second is a low volume suite designed for potent compounds in low volumes. And then in the middle is a utility area for feeding utilities out to both continuous manufacturing process trains. After API production, we have the API in-process storage area. The API in-process storage area consists of 4 vessels for the large volume API train and 2 small vessels for the small volume API train. Also in the API in-process storage area is a skid that's going to be utilized to make liquid concentrates of the API. These liquid concentrates will be pumped from the in-process storage area over into the vial filling formulation area. In the event that we need bulk powder for OSD production, we will use vacuum conveyance to move dry API from the in-process storage area into the OSD area. Next we have the sterile core vial production area. We'll start with the formulation of the vial production area. In this formulation area is designed with 3 different modes of operation. The first mode is fully continuous formulation, where we will continuously blend concentrates coming in from the in-process area with water for injection in the formulation area, and we will continuously adjust for content and pH. And then we will continuously feed that solution into the vial filling line. The second mode of operation would be a semi-continuous mode, where we would utilize 2 vessels, and we would make a 24-hour lot of liquid in one vessel to feed out of, while we were prepping the second vessel for the next 24 hours. And we would go back and forth between those 2 vessels every 24 hours. The third mode of operation in this area is the ability to take API in from external sources and produce a batch solution for vial filling. Next, we have the vial filling line. This is a commercial scale high throughput vial filling line, 400 vials per minute. We start at the top of the line up here, and we put empty vials into the machine. First step is the vial washer. After vial washing, we go through a depyrogenation tunnel. After depyrogenation, we then fill the vials with the solutions coming in from the formulation area. Once these vials are filled, we then go through the stopper station. After the stopper station, we hit the capping station, and then we finish off with a bright stock labeling unit operation. Stoppers and caps come into the machine through a RTP port, and the RTP port will be docked up with a material transfer isolator. After vial production, we go into terminal sterilization, where we will terminally sterilize our vials. Upon completion of terminally sterilizing the vials, we will then go into a automated vial inspection area. Upon completion of inspection, vials will be palletized and brought into the warehouse for short-term storage. Next, we have a look at the oral solid dosage form area. This is a future installation, and it's based on our proprietary injection mold and coating technology. Bulk API will be vacuum conveyed in from the in-process storage area into the OSD area. In addition to the API, we have a dispensary on the second floor where we will dispense excipients and utilize mass powder vibratory feeders to add excipients into the OSD line with the API coming in from the in-process area. Now we will get into a challenge associated with converting a batch process to an end-to-end continuous manufacturing process. One of the biggest challenges to continuous manufacturing of pharmaceutical products is how to define a lot, and how that lot definition is impacted by control strategy. We can define a lot by the quantity of product to be produced, the quantity of starting materials that we add to the process, or the runtime at a defined mass flow rate. Every 24 hours is a lot, for example, or some other definition that's required based on the unique nature of any given process. Continuous manufacturing typically involves longer runtimes than a batch process. In this example, we're going to assume that we are running continuous API production for 4 months. In this example, we would use a continuous API manufacturing process to produce 3,000 kilos of API. And we would call this entire 3,000 kilos one master lot of API. In a continuous manufacturing process, you are continuously adding solvents and raw materials into the process, and you are continuously removing impurities from the process. This is a simple example where we have a single solvent and a single raw material, but in reality, if you're working a real process, you probably have several solvents and several raw materials, all of which have different size lots associated with the master lot. Just as we are continuously adding raw materials and solvents into the process, we will be continuously drawing product out of the process. In this example, the product coming out of the continuous API manufacturing process would be sent directly into a drug product manufacturing process, aseptic or a sterile vial production process. Now, this process is very different from an API production process in a sense that continuous manufacturing of vials is limited with respects to how long sterility can be maintained in the vial filling line. So whereas in the API production area, it's conceivable to get the process running in steady state and run it continuously for 4 months, while in a vial production area, that assumption probably is not going to be achievable with respects to the limited length of time that sterility can be maintained in a vial filling operation. In this particular example, we're assuming we can maintain sterility for 14 days of continuous vial production. Each 14-day sublot of vials would produce 7 million vials, after which the machine needs to be shut down, disassembled, cleaned, reassembled, and re-sterilized prior to the introduction of sublot 2 for vial production of sublot 2. And this pace would continue for 4 months, and we will have produced a single master lot of API, and from that single master lot of API, we will have produced 7 sublots of drug product. A very important question to ask is, after the first sublot of vials is produced, which would be 14 days into a 4-month production window, how would we go about testing and release of drug product of sublot 1 vials, while the master lot of API is still in production for 3.5 more months? Well, we can see 2 options to this question at this point. Option 1 would be to utilize drug substance sampling prior to introduction of drug substance into the drug product process. Now, we are going end to end, so we would not be taking the drug substance out of process. We would sample the in-process storage tanks and test these samples for your traditional drug substance critical quality attributes. I put generic critical quality attributes of drug substance on the left, essentially appearance, solubility, specific rotation, ID, impurities, alkalinity, residual solvents, and water content. These are traditional CQAs that you could find on a C of A of active pharmaceutical ingredients that were purchased for use in a fill-finish operation. We also would then sample and test drug product vials for traditional critical quality attributes associated with identity, assay, impurities. Now we pick up sterility, bacterial endotoxin, and pH. So in this scenario, we're fully characterizing drug substance and drug product while leaving the drug substance in process such that we could go end-to-end of production from raw materials to drug product. The second option involves all testing to be performed on the drug product in the liquid formula phase. So all of your drug substance critical quality attributes and all of your drug product critical quality attributes would be tested on the vials that come off the vial production line. And if you combine the critical quality attributes of drug substance and drug product, you come up with a combined list of end-to-end critical quality attributes of identity, assay, impurities, sterility, bacterial endotoxins, pH, residual solvents, and water content. Now, there are benefits to both of these options. As part of our contract with the Department of Defense, we are contracted to both develop a drug master file for drug substance as well as an abbreviated new drug application for an end-to-end process utilizing continuous manufacturing. So option one, where we sample the in-process storage area powder and we fully characterize the drug substance, in addition to fully characterizing the drug product, will give us the ability to develop our drug master files for the APIs that we're going to be producing. On the other hand, if we are going to file an abbreviated new drug application for an end-to-end process from raw materials of drug substance all the way through drug product, option two may be a more suitable strategy where we sample and test drug product for the combined drug substance and drug product critical quality attributes. And this scenario would enable an end-to-end abbreviated new drug application. And with that, I hope you enjoyed a virtual tour of an end-to-end continuous manufacturing facility for the production of critical medicines that are in short supply here in the United States. There are many benefits to end-to-end production and continuous manufacturing. Cost reduction of the overall product can be achieved. We can reduce the number of unit operations due to the integrated nature of drug substance and drug product production without isolation. We can decrease the amount of solvents we use in the production of drug product due to the smaller size of a continuous manufacturing operations. We can achieve an overall energy efficiency due to the removal of costly transportation as well as lower energy costs associated with continuous manufacturing. We can reduce the footprint of the facilities required to produce drug substance and drug product when you collapse two factories in under a single roof. And we also can reduce the lead time of delivery of product from the start of raw materials to the drug product availability. All of these benefits and then some can be achieved. However, as an industry, we are going to have to work on the required solutions to the challenges that exist while converting processes from batch to continuous manufacturing. And with that, I want to thank you for listening to my presentation, and I'll be happy to answer any questions in the live sessions. Have a good day.
Hello, everyone. My name is John Lewin. I'm the Chief Medical Officer at On Demand Pharmaceuticals. It's an honor and privilege to be here with you all today to speak about our organization's efforts in moving advanced manufacturing forward. Before I begin, I want to thank the organizers of the Pharmaceutical Quality Symposium for pulling together such a terrific agenda and list of speakers. It's truly humbling to be speaking alongside each of them. Before I start, a few disclaimers to offer. One is I'm not an FDA employee. The research that I'm describing within this slide deck and presentation was in part funded by the US government, and the views and conclusions contained therein are my own and do not represent any policies either expressed or implied by the FDA or the US government. The original concept for what we are doing came from a real-world need. Our CEO, Dr. Jeffrey Ling, who is a retired Army colonel and physician, was deployed in Afghanistan and needed a particular drug that he couldn't get. They needed a medicine to treat traumatic brain injury, to manage a young soldier's dysautonomia. And fortunately, he was able to get that medicine flown in from Germany after an extended period of time, and that soldier did go on to make a full recovery. But that particular incident stuck with him. It stuck with him so profoundly that that spawned the beginning of the efforts to move medicine manufacturing closer to the patient. So what are our objectives and why are we doing this as an organization? First and foremost is to develop additional resilience and responsiveness and added health to the warfighter. We're funded by DARPA and HHS, and our activities are designed to develop new medicine manufacturing capabilities for the Department of Defense. And in particular, those activities are centralized around essential medicines. And that means, for the most part, generics. But more so than that, we see this technology and the platform development as being an enabler to devise a new approach and a new way for how medicine manufacturing interfaces with the patients. And we see advanced manufacturing in general as being a real-world solution to some of the problems which I'll describe in more detail in terms of access to essential medicines and drug shortages. So at ODP, we are under one roof, developing automated systems to produce fine chemicals and key starting materials, active pharmaceutical ingredients, automated systems for formulation, fill finish, and oral solid tablet production, and vertically integrating the entire capacity of the supply chain in a small footprint. This past year, we've been able to rapidly accelerate our capabilities by moving into a new headquarters, outfitting a CGMP suite, as I described, and a growing team of very talented professionals to move the needle forward. Our systems to date have produced over 16 different KSMs and precursors, 21 different active pharmaceutical ingredients, a number of tablets and sterile injectables, and we are rapidly progressing the platform capability in all domains. I think for many of you listening to this presentation, this information won't be new, but it's worth revisiting briefly. Our current ecosystem for how we provision medicines, in particular generic medicines, is not functioning optimally. We have a scenario where we've had underinvestment in facilities, offshoring of our manufacturing capability, all in an effort to arrive at a place where we have inexpensive generic medicines for our patients. But that pendulum has swung so far that it's produced legitimate quality issues and very challenging market dynamics, often referred to as a quote-unquote race to the bottom. The challenge is that 9 out of 10 prescriptions taken by Americans are generic. And essentially, with China as the world's largest chemical producer of fine chemicals, with 40% of the global market, and India producing the majority of small molecule APIs, also being highly dependent on China, leaves us with a scenario where roughly 85% of our medicines are vulnerable to those types of supply chain disruptions if trade with Asia were interrupted. Moreover, this current ecosystem, from raw material all the way through to finished drug product, can take as long as 300 days to make its way from beginning to end. And so that process and that time frame, leveraging old facilities and outdated activities, processes, and technology is not a system that results in the responsiveness that we're going to need going into the future. There is an increasing recognition that this is producing a national security challenge in terms of our dependency for particular medicines. I have a few examples on the screen here: ciprofloxacin, an antibiotic, nevirapine, an antiviral, and midazolam, a sedative, all just emblematic of the larger supply chain of small molecule generic medicines. And you can see here the flags in terms of the country of origin for some of the key starting materials and intermediates used in the creation of these medicines. And just with a single glance, you can see very quickly that if we were to have to make these ourselves, we would be challenged to do so because the raw materials are coming from elsewhere. Now, of course, there's good reason for that. The wages and the environmental regulations in some of those other countries are more favorable for producing a particular molecule at a reduced price as compared to the capability to produce this in the US. So we see this as a legitimate challenge. What we don't want to do is build facilities and processes here in the United States the way that they are done now. If we do that, we will constantly be pressured to have to fight against those lower wages and environmental regulations that are less stringent in those other countries in order to be cost competitive. So what that means as an ecosystem, we need to innovate and science our way to better solutions. But these problems are not new, and these problems have legitimate impact on patients, and they have legitimate impact on frontline providers' ability to care for patients. As a critical care pharmacist who has been practicing for over 20 years, I have lived the struggle of not having medicines that I needed when I needed them. And that has been going on for decades now. So here you see on the right-hand side a graph of national drug shortages. Now this graph is produced by ASHP. It's a little bit different than the shortage webpage for the FDA, both of which contain very valuable information. But you can see here, right around 2005 and 2006, we've begun to see increases in the number of new shortages every year exponentially, and those number of new shortages have sustained. Part of the additional challenges here is once a drug goes on shortage, it tends to stay on shortage for a long period of time. Now, it might be easy to take a look at some of these medicines and say, well, there are good alternatives. Like if you don't have midazolam, you could use a different benzodiazepine. And in part, that is true. But one of the serious challenges here from a frontline provider perspective is that when you switch from one drug to another, when all of your providers are used to using a particular drug at a particular concentration under particular conditions, that is a recipe for generating more medication errors and patient harm. We built a lot of safety into these systems at the front line, and having to change those systems frequently and often to accommodate a highly unreliable supply of medicines produces additional labor hours. One estimate more recently done by Vizient suggests that it's almost 9 million hours of additional labor annually and at least $360 million of annual spend by hospitals just to mitigate and manage the drug shortage process. And this includes rationing of medicines. This includes dealing with situations where up to 71% of hospitals have reported an inability to provide patients with the preferred drug, having to use alternatives. 75% of reported patients' treatments have been delayed, and nearly 100% of them have reported having to ration or restrict supply. These are common events in hospitals nowadays, and this is a paradigm that we think we ought to be able to address and fix. At ODP, we are working to miniaturize, automate, and implement the entire global supply chain capabilities under one roof. And for us, that means producing and making the equipment that run the processes. And we do that in parallel to developing our processes that either make the drug product itself, the APIs, and or the precursors. And we are implementing quality management systems across the organization with an eye towards virtual digital QA and production control at the point of care. And the way that we do that is we implement rapid prototyping capabilities and very fast cycle times to evolve our systems quickly to make them GMP capable. We leverage advanced automation and programming capabilities of processes and systems, and leverage our modularity and plug and play capabilities of our unit operations to give us ultimate flexibility between switching between different products and processes. As we develop our processes in terms of either synthesizing the API, precursors, or formulating and creating the final drug product, we leverage quality by design principles, and we implement state-of-the-art PAT and advanced process controls to develop the necessary requirements to drive towards real-time release testing. Key components of these activities include implementing shared services across the continuum and the team members in each of the paradigms I've listed above, and that includes IT infrastructure and cybersecurity, project and lifecycle management across each of the efforts that we are engaged in, and warehouse storage and distribution, both for components, device, raw materials, and products. Implementing a novel quality management system that's going to result in high transparency downstream for frontline providers, both on the process and the device, is also something that we are passionate about and working towards. And then implementing quality control capabilities on both the product, the process, and the device as well. At ODP over the last year and a half, we've been able to rapidly progress our organizational capabilities to support the rapid development of our platform technologies. And one important way in which we do this, of course, is attracting and retaining some of the very best talent. And we've been successful in doing so, gratefully, and harnessing that talent through novel multidisciplinary integrated workflows. And so this includes, just as an example, as you can see on the screen here, embedding workflows across chemistry, engineering, data science, and statistics, along with quality, science, and qualification and validation activities up and down the continuum, as long as imparting best practices from a regulatory perspective and manufacturing sciences perspective. Within our facility, we quickly developed an R&D analytical lab to support our chemistry and formulation development activities, and this includes, of course, developing new, highly intensified processes on small automated systems for old drugs, so often requiring development of new chemistries, leveraging state-of-the-art techniques to arrive at the most efficient and effective process that could then be automated and programmed and reproducible, along with quick changeovers between processes. So these R&D, chemistry, and formulation activities all come together in our translational lab and integration space to develop the programmable recipes on our systems for implementation. We have a state-of-the-art machine shop, and this allows for rapid prototyping of devices and unit operations on our manufacturing equipment. And it also enables us critically to do rapid cycle turnover and improvements to develop those systems and capabilities. And then lastly, as of the current date of this presentation, ODP is nearing completion of build out of our GMP QC laboratory capabilities and a state-of-the-art GMP suite where we'll be leveraging our technologies to rapidly progress the advancement of these platforms. Through the enabling capabilities described on the previous slide, we have rapidly advanced our systems and processes across the platform. And when I say platform, I'm referring to our Pharmacy on Demand platform, which broadly can be described as miniaturized, automated, programmed molecular systems. And this has included bringing on new precursor manufacturing systems and capabilities, as well as API production systems and new purification systems and capabilities. But embedded behind all of this across the platform is really an enhanced modularity an ability to produce multiple medications, and an ability to allow for quick change between the production of those medications. We see these foundational elements as being drivers of a U.S. source material supply chain that is resilient and responsive to surges in demand. And this is one of really what's going to require a ecosystem-wide effort to combat this race to the bottom through using advanced manufacturing techniques and progression and innovation on the science and technology. What I'd like to do for you on this next slide is describe to you a potential for an innovative, patient-centric, more transparent ecosystem for how medicines are provisioned to patients. So to draw a contrast to the current state, we have described previously how we have an unreliable current process that spans the globe, that's dependent, at least for the US, on foreign adversarial sources of material and a supply chain that has a lot of opacities to it. And that supply chain eventually does result in a finished drug product that makes its way to a pharmacy. Now, at the sharp end, in terms of patient care, there's not much that we know about the ingredients that go into this. And there's very little that we know about the quality of the manufacturer or the systems that have produced the material. Now, this medication, of course, is typically manually compounded into something that can then be administered to a patient under most conditions. And this manual compounding process can also be fraught with challenges and errors. Now, there's, of course, a lot of variability in how compounding is conducted across the continuum. And a lot of that's driven by the fact that compounding is overseen, for the most part, by states. Some are more stringent than others in terms of the quality standards that they require. And of course, some of those quality challenges have driven some serious adverse effects in terms of things like the New England Compounding Center and increased quality risks. And then that, of course, has driven the need for 503B outsourcing facilities to mass compound these materials in a way that hospitals and health systems can then utilize them efficiently and safely to manage patient care. The challenge here is that this system has not met the needs in terms of what providers need to care for patients. And that was clearly shown during, especially the early phases of COVID. I can attest personally to having challenges with obtaining the medicines I needed for patients, and know of multiple health systems that had to go so far as entertaining standing up non-sterile to sterile compounding operations just to get medicines to keep their patients comfortable on the ventilator. So this is a system that did not meet the demand during the pandemic, and it's a system that produced a lot of drug shortages even prior to that. Now, since we're talking about advanced manufacturing techniques and pushing the envelope forward, I'd suggest a future state, whether it's in the United States or across the globe, we have a capability to produce the materials we need at the point of care, leveraging some of the innovations I've described previously. This can produce a much more resilient and responsive supply chain. I also think that we need to move manufacturing closer to the patient. We need to change the relationship between how medicines are made and the frontline providers that use them. Why can't we move automation and quality standards that meet cGMP to the point of care? We can leverage these two new techniques to take a giant leap forward. This is going to require innovations in automation, control, implementing real-time release capabilities, and of course, a number of other factors in terms of understanding of the environment in which these systems run and maintaining their security. But by leveraging these new advanced manufacturing capabilities across the ecosystem, we can begin to define a more patient-centric way for manufacturing of essential medicines and generic medicines and how they get to their patients in the most ready-to-use form with the highest level of quality and control with ultimate flexibility for the frontline providers in order to meet their patient care needs. Now, at ODP, one of the ways we see ourselves revolutionizing this process is through a multi-pronged approach. And that approach requires a deep understanding of the current challenges. And so that's why we do spend a lot of our time developing predictive insights across the reaction space for small molecule chemistries, as well as understanding the intricacies of pricing suppliers and customers and markets, and leveraging that big data to develop those predictive insights. That in turn then drives our focus in terms of developing programmable chemistries and automation, new process analytical technologies, so that we can leverage our internal capabilities to go faster, to develop more resilience and adaptability in our responsiveness to need. And then lastly, by putting the medicine manufacturing capability at the point of care with full automation, with integration into those health systems and records, that gives us an ultimate ability to provide personalized medications, enhance safety, and ensuring, importantly, quality in every dose with limited variability. And we can do this by, and at the same time, drive cost savings across the health system paradigm. This distributed manufacturing capability, I think, has been pushed forward to the point where there's broad recognition that having very, very focused areas of manufacturing leads to disasters, whether it's natural disasters or man-made disasters. Having everything located in one place produces a challenge. By distributing our capability and embedding flexibility across our systems, that gives us an ability to respond to threats that are both known and threats that are unknown as well. Of course, in order to apply and implement 21st century manufacturing technologies and techniques in a way that is going to optimize patient care will take a team effort. It's going to take an ecosystem working together across regulators, policymakers, manufacturers, financial institutions, as well as the technology and healthcare sector. So finally, let me leave you with this vision for the world, wherein we have an ecosystem that can provide medicines flexibly at the point of care. And that ecosystem and that technology results in resilience and responsiveness for those that are protecting us. It results in access to quality medicines, every single dose, all around the globe, including areas that are underserved, that have diseases that have been treatable for decades. And it results in medications that are affordable and accessible for all. And it meets the needs of our healthcare providers so that they can optimally take care of patients. And if you're listening to this symposium and all the fantastic descriptions and lectures herein, it's presumably because you're a mission-driven individual and you are excited to be in the business of providing quality medicines to patients. So let's all think about how we can continue to innovate our ecosystem and push a vision forward that makes sure that we address all these challenges that we have. Thank you, and I appreciate you listening today.
Thank you for those excellent presentations. Our last three speakers will now join us for a panel discussion to answer your questions. If you haven't yet had a chance, please enter your questions into the Q&A pod. We will start with a question for Dr. O'Connor. Can you talk about other advanced manufacturing technologies that have been used by the industry?
Yeah, thank you for that question. I tried to give some examples during the presentation today, but there are many other examples, mostly some continuous manufacturing examples, both on the small molecule drug product side, where we do have existing research on loss and weight feeders and other areas, continuous manufacturing for biologics. We recently had a research impact story on viral safety research. So those are a couple of other examples. Thank you for the nice question.
Thank you, Dr. O'Connor. We do have another question for you. Can you describe how research projects in OTR are selected for investigation?
Yeah, thank you. We try to align our research program to the needs of OPQ and center. So it's what kind of technologies are we seeing through the application assessment process? So we try to align very closely with what we're seeing for the emerging technology team. We have a very close interaction there. So the questions that are being posed by sponsors can, when needed, generate new research projects to address that. And we try to be forward-looking. Adam spoke about developing a framework, right? And it's going to be based on science. So we try to be also proactive in identifying where we see gaps in the future and try to proactively address that by selecting research projects that will give us the data and information and knowledge that we need to formulate science-based processes. Thank you for the question.
Thank you for responding. We have another question for you. What is OPQ's plan for the pilot plant to support advanced manufacturing?
Yeah, thank you for that question. It's something we're very excited about. The pilot plant is designed as a modular space. And while we looked at different things individually so far in our research program, this will really give us a space and a facility that allows us to integrate things. And that could be integrating unit operations. You've seen some examples of that today. Integrating measurement systems with the manufacturing process, integrating models with equipment, advanced process controls, and really give us the capability to kind of do that kind of projects. The facility will also have a dedicated training space, and this will really help with workforce development, preparing the FDA, a large pool of people at the FDA to be ready to assess these new technologies, help develop policy around them, and really support all these initiatives you heard a little bit about today. Thank you for the great question.
Thank you. Going to our next question, it is again for you. How does OPQ decide which extramural projects to fund?
Thank you for that question. Whether it's submitted through a grant opportunity or through the regulatory science broad agency announcement, all proposals for projects go through a technical review. So that means SMEs throughout the agency are assigned to review that, and they're looking for both the scientific merit of the project, the team is the right team, the right infrastructure. But another critical piece is the regulatory relevance. So, is it addressing a question that we need new science to help us answer to do our job, whether that's an assessment, a policy. So that's another critical piece because we often get a lot of great science proposals, but we're really looking to pick projects that are going to address kind of mission critical questions for us to help us either move an application assessments or policy development. Thank you.
Thank you very much for your responses. Let's move to a different speaker. This question is for Ernie Penachio. And the audience would like to know, how is the continuous sterile production system scaled? Is it based on vial filling capacity and then API production is scaled down to match? What is the pinch point in keeping things running continuously?
Thank you for that question. We would start with developing an API production process that would meet an annual demand that had been requested. Then we would take a look at the vial filling operation and scale the vial filling operation speed up and down between, let's say, 100 and 400 vials per minute. And the third degree of freedom we would have is an accumulation tank that sits between drug substance and drug product manufacturing that will allow us to accumulate material and compensate for differences in throughput. And thank you for the question.
Thank you, Ernie. Another question for you. How will the sampling of continuous manufacturing of the API and FP occur?
The sampling will occur on the drug substance side in the accumulation tank. As the vial filling process starts up, we would utilize a beginning, middle, end sampling approach to the API that's being continuously formulated. Additionally, we could use, in the accumulation vessel itself, we could use a top, middle, bottom sampling approach, again spread across the length of the vial filling operation. From a vial filling perspective, those lots in general would be sampled for each sublot associated with terminal sterilization. Each terminal sterilization run, we would sample an adequate number of vials and test them for sterility as well as uniformity. And thank you for the question.
Thank you, Ernie. Let's move on to Dr. Lewin, a question for you. A fundamental problem underlying the attempt to reshore drug manufacturing is the need to source raw materials from overseas. Do you see any solutions to that issue?
Thank you for that question. That's a great question because that is a big challenge, I think, for all of us. One approach that we're taking at ODP, as I described a little bit, is doing some of the chemical mapping and the types of reaction spaces we will need to go very far back in the synthetic pathway to raw materials that can be sourced here in the U.S. So I think it's going to take, multiple pronged approach to solve that challenge. But one way to do that, and one approach that we are taking, is through the modularity and automation in our systems, is to back up the chemistry far enough where we can get a starting point that is locally sourced as a means to secure the supply chain completely. Thank you for that question.
Thank you, Dr. Lewin. We have another question for you. Have you determined any geographic areas of the U.S. that are best suited for an ODP installation? Do you have a pilot in place already?
That's a great question. We do have a pilot in place. Our facility in Rockville, Maryland is our initial starting point, where we're building out our capabilities. We would plan to expand beyond that. And I will tell you that we are doing a lot of modeling activities to determine what that type of networked distributed capability would look like, how that would need to be resourced, and what the outcomes of that might be from an optimal distribution perspective. But you raise a good point. I think it's a point that another person raised as well in terms of a question, is there are significant geographical disparities in how we move our medicines around the country. And the recent pandemic illustrated that quite well, where you had large amounts of supplies in some areas of the country, or consolidated in some hospitals or health systems that had larger purchasing power. And you saw scenarios where smaller facilities or other more rural areas could not access those medicines. And so that is a weakness of a highly centralized model. And a way to strengthen that is to distribute the manufacturing process a bit further. It helps to get to solving what's frequently called that last mile problem when we're talking about having an agile response capability. Thank you for that question.
Thank you. And we do have another question for you. How much operation interaction is required for normal operation of the continuous manufacturing facility, and where are the main interactions occurring?
Is that a question for Ernie?
That was a question for you. Would you like to turn it over to Ernie?
Ernie, can you help with that one?
Sure, I can jump in on that. With respect to interaction of the equipment, every process I find is different. When it comes to the continuous manufacturing, some processes run very smoothly, and our API production is fairly standard. You have reactions. You have crystallizations, filtrations, things like that. I find that the most challenging part and the most interaction with the equipment is around crystallization, where it can be difficult to get into a steady state, and as we're moving materials in and out of the process, we can get process drift, and that's a spot where we can see a lot of interaction. There are other parts of the process that run very smoothly and essentially take very little monitoring, very little interaction with the equipment. I hope that answered the question. Thank you for the question.
Thank you. Dr. Lewin, we have another question for you. What do you think the biggest barriers are to adoption of new technology?
Well, I mean, there's several, right? Part of the challenge is if you're the first person to move into a space, you do take on a lot of risk as an organization from a regulatory perspective in terms of pushing the envelope. Sometimes it's easier to be second or third in line, if you will. That presents its own set of challenges. The great thing is, though, with this symposium today, the agency has been, very proactive in developing, collaborative relationships through the Emerging Technology Program to help facilitate, very strong discussions with industry partners to help accelerate this process. That definitely is one of the key barriers, and it should be, right? Because we do got to make sure that any new technologies we deploy, it is our responsibility to make sure that they are producing product that is safe all the time. And so that is one way to grease the wheels. I think, another opportunity is to continue to think along what some other incentives might be to encourage industry to push this envelope forward, particularly when you're talking about folks that are making essential medicines where the margins are very narrow. The hurdle to jump into and apply the latest and greatest technologies to those particular medicines is that much greater. Thank you.
Thank you. And that's all the time we have today. We will now hear closing remarks from the Director of the Office of Pharmaceutical Quality, Dr. Michael Kopcha. But before I turn it over, we would like to remind you to please complete the surveys and to sign up to receive information about upcoming events via SBIA, LinkedIn, and email updates. Recordings from this two-day conference will be posted within 2 weeks to www.fda.gov/CDER-Learn. And we will now turn it over to Dr. Kopcha for closing remarks.
Thank you so much. Yeah, I just want to reiterate that, we do appreciate the feedback, so please do take the time to provide us with your feedback so that as we plan our next meeting, we will be able then to, kind of adjust accordingly and look for some topics as well. So what I want to mention is that when I kicked off this event yesterday in my keynote, I shared several lessons I learned over the past 2 years as the director of the Office of Pharmaceutical Quality, or as we like to call OPQ. However, I did leave out one very crucial lesson that I do want to share with you, and that is the importance of gratitude. And I just want to stress that I am grateful for how an event like this brings different kinds of people together. Though we may be different, we do come together from all around the world. I do believe that we are all intent on meeting the same goal, and that is that assuring quality medicines are available for patients as well as for consumers. When I say we are different, I don't just mean that the audience is made up of a diverse group of people, but that even the speakers came together from many different parts of the agency to rally around quality. Because let's face it, quality, in my eyes, is one of the 3 components when we talk about drugs. We talk about safety and efficacy, but quality is also a very key piece of that, and we need to keep that in mind. Yesterday, we heard the keynote from our acting commissioner, Dr. Woodcock, who kicked off our event with a very powerful keynote, which emphasized her personal passion for innovation. We had a panel of leaders from across the FDA yesterday as well. One of which I participated in, which was gave us an extended live discussion yesterday. So I'm always appreciative of those individuals that join us from around the agency to add and enhance the message that we're trying to get across as in regards to quality itself. So I'd like to first personally thank Don Ashley of CDER's Office of Compliance, Teresa Mullen, who's CDER's Associate Director of Strategy, and Elizabeth Miller of the FDA's Office of Regulatory Affairs for their thoughtful insights and for their willingness to participate yesterday, because they are all very busy people. I do appreciate them taking the time to really share in the discussion along with me. I love how this event also brings together people from my own organization together to speak on topics that they too are so passionate about. So for some, this was their first time speaking publicly on these topics. For others that are more seasoned, and perhaps none more so than OPQ's Brian Hasselbach, who, as you had heard yesterday, has served the agency for over 3 decades. So that's 30 years of being a public servant. I do want to mention, though, that Brian will be retiring shortly. I'm grateful for the knowledge and dedication that he brings to our organization every day. It goes without saying that he will be sorely missed by all of us, and we collectively wish him all the best as he does retire from federal service. So I did want to mention that because I know a lot of you may be familiar with Brian. He's been around obviously for a long time, but he brings a lot of value in the discussions that we have, not only within OPQ, but within CDER, within the FDA, and he's really a sought-after individual for his opinions and kind of the the knowledge that he has and that he's gained over those 3 decades. We've even invited speakers from outside the agency so that you could hear about manufacturing innovations and better understand how they are being implemented as well. So we're not only focused on what we're doing, but we really like to bring in an outside perspective. I appreciate these important stakeholders for dedicating their time to be part of this event as well. Now it is very easy to underestimate the amount of work that goes into planning and executing an event that is run as seamlessly as this one. So I do need to give a special thanks to Brenda Stoddart, Jeff Kelly, and everyone that supports SBIA in running these symposiums. They always do an extremely professional, well-organized, well-coordinated, well-executed meeting, and I can't thank them enough for doing that. And again, sometimes they go unnoticed because things just work so seamlessly. People don't realize the amount of time and attention and professionalism that needs to go into that. And these individuals are second, and I really truly mean this, second to none. Finally, a symposium would not be possible without an audience, so I'm grateful to each and every one of you for taking the time to be here with us virtually over the last 2 days. Your questions, your insightful questions and comments, and our interactions are really the value driver of a symposium like this, so I'm really glad to see that people take advantage of the opportunity to interact with all of us. I'm already looking forward to engaging with you again at the next symposium, which will be in 2023. Yes, you did hear that right. It is 2023. We do these. We do the symposium once every 2 years because we do want to make sure that we really have a number of good topics to talk about, and it gives us some time to actually plan it and plan it in good detail and make it a good, meaningful symposium, not only for us but for those that attend as well. In the meantime, let's continue using innovation to give patients and consumers confidence in their next dose of medicine. So again, I thank you for the privilege of your time and for joining us, and I look forward to hopefully most, if not all of you, returning in 2023. So with that, I will then close the meeting. Thank you.